Hello SBI3U. This is video 3. It's about DNA and the code of life. Here's our learning goals. We're going to talk about the structure of DNA, just the basic kind of building blocks of it, the base pair matching. So this is called Chargaff's rule. It's just named after some dead white guy. We'll talk about the double strand structure of DNA. So the semiconservative replication of it. You'll learn what those words mean. And then we're going to get into the actual meat of the lesson, which is mutations. So all of this background information is really just to explain why mutations occur. So that's going to be the keep in mind up here the entire time, right? We're learning all this just to explain how mutations occur and how UV and certain chemicals can cause mutations. So you might have heard of like carcinogens. We're going to try to explain the biology behind that. So jumping right in, let's talk about our current topic, which is the structure of DNA. And we are going to just jump right into it. So DNA stands for deoxyribonucleic acid. It's the thing that carries genetic information. You know about this. It's in the nucleus. And in bacteria, which don't have a nucleus, it's just in the bacteria. And certain viruses carry DNA, and then they can use that DNA to make cells do stuff. Mind you, not coronavirus. That one is an RNA virus. So, chemical composition of DNA. This was a guy, Phoebus Levine, which is a wonderful name. Like, next time you're making like a fantasy character, like some kind of archer or something, name him Phoebus Levine. Like, great name. He figured out that DNA has these three main components. He was able to separate the big giant molecule of DNA into a sugar, a phosphate, and a nitrogenous base. And he just did this with chemistry, some kind of chemical stuff. And he found out that the number of sugars was equal to the number of phosphates and was equal to the number of bases. So he came up with this. He came up with this idea for DNA where there's always a phosphate attached to a sugar attached to a nitrogenous base. Now this negative charge is going to get very important in tomorrow's lesson, where we're talking about DNA technologies like gel electrophoresis particularly. We'll see that in gel electrophoresis they use electric charges to make the DNA move through a membrane, and it moves because of these negatives. We'll see that again tomorrow, just highlighting the fact that there's negative charges on every DNA molecule. So this is the basic unit of DNA. This is one phosphate, one sugar, one base. And then the code is made up of the bases. There are different bases. So here are the next piece is about the different DNA bases. So just update, the four DNA bases. The four DNA bases, they are A, G, T, and C. That's adenine, guanine, thymine, and cytosine. Adenine, guanine, thymine, cytosine. The A and the G are big, and the T and the C are small. The big ones are called purines, and the small ones are called pyrimidines. Which is always annoying to me because purine is a smaller word than pyrimidine. Like it comes from a chemical definition, like purine is a specific chemical like ring structure and pyrimidine is a different specific chemical ring structure, but it always annoys me that the small ones have the larger name and the large ones have the smaller name. Here they are, right? You can see guanine and adenine are bigger. They have two rings. They're purines, and thymine and cytosine are smaller. They're pyrimidines. One way to remember this is purines are giant, so PAG, purines are giant. So purines are the adenine and the guanine, and pyrimidines are small, which there's no mnemonic that I know of. It's just purines are giant. So those are the four bases. Notice there's a lot of nitrogen on them. That's why they're called nitrogenous bases. There's a bunch of nitrogen in DNA. Here's another view of it where you see the big purines and the little pyrimidines, but you know, it's just the same structures. Same structures, just in a different arrangement here. So A and G, T and C. So that, I believe, is, yeah, that's the end of our first little thing. This is the structure of DNA. You've always got the ribose, deoxyribose, always there. You've always got the phosphate, and then this will change depending on which base it is. It could be A, T, C, or G, and they all attach to this nitrogen here. So it's this nitrogen that it attaches to, that little guy right there. So there's the structure of DNA, discovered by that guy with the cool name, Phoebus Levine. Yeah, great name. So jumping into now the base pairing. So if we review the base pair matching, which is Chargaff's rule, this is going to be a very, very quick part of it. So let's change current topic. Chargaff's rule. Chargaff's rule of base pairing. So take that away. Let's actually just talk about Chargaff's discovery briefly over here on my whiteboard. Oh man, I have a lot of stuff written on these whiteboards. There we go. So let's talk about Chargaff's discovery on the whiteboard here. Basically what he did was he kind of he figured out how to find the proportion of the bases, and he analyzed a bunch of different animals. So this isn't actually his ratios, but the idea is there. He kind of analyzed the A, T, C, and G content of the DNA of different animals. So maybe he did like human, maybe he did dog, maybe he did cat, maybe he did, I don't know, fish, mosquito, whatever. Maybe he did like E. coli. Everyone likes E. coli. So he kind of analyzed the percentages, and he found, once again, these aren't his ratios, but it's representative. Maybe he found in humans that it was like 13% A and 13% T, and it's 26, so 74 divided by 2 is 37%. So he always found that these percentages matched, and he found that it was different in different animals. So like dogs might be 16%, right? 16%, and then 34% C and 34% G. And like cats might be 8%, and 42%. And so on. The pattern continued. He noticed that that the amount of A and the amount of T was always very similar, and the amount of C and the amount of G was always very similar, like within a percent of each other. But the C and the G were different between each other. And he also noticed that different animals had different proportions. So this suggested to him two things. The suggestion was two things. The inference, I guess. The inference was A matches to T somehow. A and T match. And C and G match somehow. And number two, this provided proof that different organisms have different DNA. He did more than that. Like he tested different humans. Like he tested different people, and he saw that they had slightly different proportions too. But not by much, but enough for him to say, hey, maybe this is the actual molecule that's transmitting information. There were other people that did much more interesting studies about that, but this was part of the proof as well. So that's Chargaff's thing. I think he got a Nobel Prize for it. I'm not actually 100% sure. Let me pause the video and I'll check. Yeah, he didn't get a Nobel Prize, but they were still wondering about the DNA thing. So so this was part of the proof for DNA. So that's Chargaff's thing. Yeah, let's jump back to the note. So that's Chargaff, Erwin Chargaff. That's commonly called Chargaff's rule down here. So next topic, if we look at our thing, is the double strand structure and the semi-conservative replication. So pulling that away, let's talk about double-stranded DNA, semi-conservative replication. So Chargaff did his thing in the 40s, 1940, and it was a big deal. But even bigger than that was Watson and Crick, 1953, who developed the actual model for the two strands. They couldn't have done it without Rosalind Franklin, who actually took the X-ray picture of the DNA and saw two strands in a corkscrew. So what this is looking at, this here is, if we had the kind of corkscrew of DNA, this is looking at it from the front. And you can see, like, if I get my finger there, and there's like that line between the corkscrew, that's what you're seeing down here with the X-ray diffraction. Yeah, so Watson and Crick used that picture and were able to add it to their current model of DNA and worked out that it's two strands that are running helixed around each other, except they're not like this, they're actually in opposite directions. They're like that to each other. They worked out that the backbone is the phosphate and the sugars, and the bases stick in. Other people thought maybe the bases stick out and the backbone is up and down, or there's like three that spiral around each other and stuff. These guys came up with the real, the best model for it. They also worked out why thymine always binds with adenine and cytosine always binds with guanine. It's based on the number of bonds, hydrogen bonds, which are interactions that these things can make. That thymine can make 2, and cytosine can make 3, and adenine can make 2, and guanine can make 3. And so that's how they figured it out, and that was their model. Right, so the sugars and the phosphates make this backbone, and then the base pairs hang in the middle. And I'm sure you've all seen these ladder diagrams of DNA where it's like these bases that are like this, and they're twisted around each other in the helix, like that, and just moving up and down this ladder like that. It's a very common view of it. One thing I'd like to point out is that the helix always goes the same way. The helix for DNA always goes as a right-handed helix. So this is how you can see the DNA helix is right-handed. So you hold out your right hand. This is my right hand, and if you twist the fingers, imagine it's like a screw, like a corkscrew. It would rise in that direction. Left-handed helix would go the other way, and this is also kind of how screws work. Screws and bolts always go in the same way, lefty-loosey, righty-tighty. Same thing here, lefty-loosey, righty-tighty for DNA. So DNA corkscrews just like a typical screw, and sometimes you'll see DNA in diagrams like on TV shows and stuff, and it's going the wrong way, and all the biologists cringe when they see that. So yeah, that's that. Start from current slide. Yeah, so that's the basic structure of DNA, but we got to talk about this stuff called semi-conservative replication, and that was a huge discovery in DNA. This is also often called one of the greatest biology experiments, one of the most beautiful biology experiments ever done. So there were three kind of thoughts on how this big long helix, so you see this helix, how this big long helix would replicate itself. The first idea was that it would just split, and then each split one would make a new like, so like you'd have the two, then they would split, and then each one would make two new strands. Right? That's called semi-conservative because you're conserving half of the original strand. This is the original strand, and then you're conserving half of them. And then you're making a new one on each one, so semi-conservative. The other option would have been conservative, where it's almost like you've got your two like this, and then like somehow it would make a second strand and a second strand, and then they would kind of, these two would go up this way and these two would go down this way, like that. Like it's easier to see on the diagram here, right? That it would conserve the entire original strand as like an original, and then there would be an entire copy in the second cell when the DNA would replicate. And then there was the other option of dispersive, and dispersive would mean that the DNA would copy itself in chunks. It would just kind of like unwind a chunk of DNA, copy it, then unwind a second chunk of DNA, copy it, and then just kind of bring together the thing. That was based on the fact that the turns in the helix, they thought maybe like each certain number of turns would just get copied before the next turn got copied, before the next turn got copied. And they thought that was because of, like, if you tried to copy too many of them, it would twist too much, stuff like that. Of course, it turned out to be semi-conservative, and the experiment that did that was incredibly cool. This is the Michelson-Stahl experiment. And what they did was they took N15, which is, normal nitrogen is N14, but there is a type of nitrogen that has one extra neutron, and it still acts exactly like nitrogen, it's just a little bit heavier. And they took E. coli and they raised it in an environment where all the nitrogen was N15. So that E. coli had DNA that had only nitrogen 15 on it. It was like heavy DNA. So compared to normal DNA, that DNA was heavy. It would be lower down in their test tube. And then N14 DNA would be lighter, and it would be up here in the test tube because it's just a density thing when they extracted the DNA. And they tracked it, they pulled out DNA from their E. coli every 20 minutes because E. coli double in number every 20 minutes. They have to replicate their DNA every 20 minutes. And they got this pattern, which to them proved that it was semi-conservative. It was a very, very, very clear pattern of semi-conservative replication, which takes a little bit of explaining. So let me jump over to the document camera and I'll explain why this proved it. So let's talk about the can't get that out of the way. That's kind of annoying. Well, whatever. You can see me. It's cool. Let's talk about the three possible methods and what that would mean for their experiment. So the first method would have been that you have double-stranded DNA. Right? I can't draw the DNA helix. Sorry, I'm a bad biologist. That would split, right, and become half and half. Right, that's the semi-conservative option. So that would be the first split. And then these ones would all split, so you'd end up with 1, 2, 3, 4, right? These would split. And then you can't make new heavy DNA. So the red was the heavy DNA. But you can make new light DNA. So each strand would get new light DNA attached to it. So what you would see in the first generation was all heavy. All of these would start with all heavy because that's what they started with. Then it would be all kind of medium. All medium. Then you'd have 2 light and 2 medium, and no heavy. Right, no heavy. Right, all heavy. All medium, no heavy, because they're both just medium. Then you'd end up with 2 mediums and no heavy. And then it would happen over and over. You'd expect the amount of medium to reduce because you're increasing the amount of light DNA. And that's what they actually saw, right? This is what they actually saw. If we look down at our experiment down in the corner here, down here, you can see that it starts all heavy, then it goes all medium, and then it splits into 50-50, and then the proportion of medium goes down over time. Let's explain why it wasn't the other ones. So if it was conservative, what you'd end up with is you'd start with all heavy, then you'd keep a heavy one, and you'd have a light one. So one would stay all heavy, and the other one would be 50% light. Right, 50% light, 50% heavy. And then the next one down, you would still have the one heavy, and then each of these would split up, and you'd have then 25% heavy and 75% light. But you'd never have a medium, never ever a medium, because the heavy would always stay together. And just by the fact that they had a medium DNA tells you that was wrong. So never a medium. And then the random one, right? The random one, you would expect like a spread, right? It would be like, kind of like that. So the first step from all heavy would immediately go to all medium. All medium. I can't spell medium. There we go, all medium. But then the next step down, right? What would happen is you're never going to get new heavy DNA, but you're going to get new medium DNA. New light DNA. So eventually what you'd get is less and less of the heavy stuff and more of the light stuff. So what you would expect to see there is as time goes on, the little test tube thing, right? It would start all heavy. And if this was all light, right, it would start all heavy, and then it would, with successive generations, first it would go to the middle, and then it would go to the middle of the middle, and then it would go to the middle of the middle of the middle, and then the middle of the middle of the middle, and the middle, like that. It would slowly like creep towards the all light, but that's not what they get. They got this incredibly obvious separate band pattern. So they knew that it was semi-conservative. Really, really cool experiment. Also didn't get a Nobel Prize. Don't know why. Meanwhile, Watson and Crick did get a Nobel Prize, but Rosalind Franklin didn't. The reason she didn't was she died. They don't give Nobel Prizes to dead people. Anyway, yeah, so these guys, Messelson and Stahl figured out that it was semi-conservative in one of the greatest experiments in biological history. And this is how they proved it. So this shows the same thing, the predictions, what it should have looked like, but conservative is the only one that matched. Sorry, semi-conservative is the only one that matched because you saw that medium band. Okay, next thing in our thing is mutations. So problems in DNA replication. So mutations. Point and frame shift. So mutations that are specifically point and frame shift mutations. So let's jump into that. The first one is point mutations, and basically remember Chargaff's rule. Chargaff's rule says that A pairs with T and C pairs with G, but and it's based on Watson and Crick's work. That was based on how many hydrogen bonds, these little hydrogen like interactions they can make. But sometimes you can get a little bit of a weird pairing where it's almost kind of a good enough fit, but not quite. It's like when you're like making a puzzle and a piece kind of goes in and you're like, maybe. And that's called a wobble fit. So T and G can do a wobble, and C and A can do a wobble, which is, backwards to the normal thing. T normally binds with A, and C normally binds with G, but you can get these wobbles. So every so often, when the DNA is replicating, the wrong one will just end up there because of these wobble pairings. So that's called a point mutation. It only changes one base out of the entire DNA genome. So most of the time it's not that bad, but sometimes it can be very bad. For example, sickle cell anemia is a genetic disease that is one single base pair that changes in one single gene. Much more serious is insertion and deletion. That happens when the DNA kind of like makes a little loop, like a little bubble as it's replicating. And then sometimes that means that depending on which strand, because it's semi-conservative, one strand stays the same and the other strand gets copied. Depending on which strand has the little loop in it, if the one that's newly getting copied loops out for a second, you end up with an extra nucleotide, so like an extra thing in the code, and that can throw off everything. If it's the template, if the one that's getting read loops out, then that T is lost, right? It never gets copied in the new strand. So you can gain one or lose one, and this is how like new DNA actually can get into the sequence. This is much more serious because it's actually changing the length of the DNA. So if it happens in the wrong place, it'll change the gene completely. It's interesting. So those are mutations, simple mutations. You can have these weird pairings, and you can have these frame shifts. And the last piece is mutagens. Mutagens. So this is putting it all together. All right, that's today's lesson. Biology behind mutations. We're going to talk about the structure of DNA. We're going to talk about the base pairing rules. We're going to talk about the semi-conservative replication. This part was the semi-conservative replication and the base pairing rules. And now we're going to talk about the structure itself. So here's a diagram of DNA in the real kind of conformation of it. Here you see the bases sticking out. These little guys are the phosphates. And the bases are sticking out, but then this here doesn't look right? There's this big chunk here that's kind of like normally it's like this, but it's kind of like out a little bit now. You see how like the DNA is kind of like out, where normally it's like a nice spiral like this. Now it's kind of like bowed out a little bit. So this thing is kind of putting strain on it. When this gets copied, this could maybe cause a bad copy error, and that stuff is this stuff, benzopyrene, and it likes to attach to this guanine. So you see this flat structure, that's actually there in 3D, and it's messing. It's inside the DNA. It's like kind of messing it up. This benzopyrene stuff is found as a component of cigarette smoke. So when you hear that like smoking damages DNA, this is one of the culprits. This is one of the things that damages DNA when you smoke. And you know, if it's bad enough, it can lead to this kind of thing, where that's going to stick out when the thing tries to copy itself. The other thing that can damage DNA is UV light. So UV can come in and actually kind of make the thymines attach to each other. So like it'll make these two attach to each other, and it'll do the same kind of thing. It makes a, you know, I'm going to put this over here. It makes a little kind of lopsided thing on the helix, and then when it's time to copy it, that can kind of get messed up. You know, a very common way for this loop to occur is for those to get crossed, and then you get a bad copy, and then you end up with a mutation. Every so often you might get a good mutation, but the more commonly it's a bad mutation because it's already, you know, the genes are pretty finely tuned at this point. Speaking of, there's one last thing here, and that's why aren't all mutations just absolutely harmful to DNA? Well, just a quick little thing to say that this red slice of, so this is all the DNA, and this is all the different like parts of it, and only 1.5% of the DNA actually codes for any kind of protein, any gene type stuff, and the rest of it appears to just be junk. We're analyzing it, like biologists are analyzing it right now, and they're finding more and more that the miscellaneous unique sequences actually do stuff, and the miscellaneous heterochromatin actually does stuff. But a lot of them, especially these introns, those are literally just chopped out of the DNA when it's being used to make a protein. So those are useless. These guys are like, they're called jumping genes. They just jump around, and they're also pretty much completely useless. So a solid 45% of the DNA doesn't do anything, but it's copied every time anyway, because that's all that your cells know how to do. That's the end of our content. So we talked about the structure of DNA. We talked about Chargaff's rule. A matches with T and C matches with G. We talked about the double strand structure of DNA, where you've got the backbone and the bases all stick towards the middle, and it rotates up in a right-handed spiral, the same kind of spiral that screws and bolts have. And more importantly, we talked about the experiment that proved semi-conservative replication. So that's a good experiment to know in terms of how they proved it. And then we talked about how different mutations can occur, so point mutations and frame shift mutations. And finally, we talked about how different chemicals and different things, so like chemicals can actually go in there and mess up the structure, and UV radiation and other kinds of radiation can go in and actually mess up the structure as well. That's it. Thanks for paying attention, and I'll see you guys in class.